I'm Ron Davis, professor of biochemistry and genetics at Stanford University, and I am the director of the Stanford Genome Technology Center, which is a multidisciplinary center. What I thought I would show you is an area that I think will be very important in the future. And it's been going on for a short time, but I think it's going to greatly expand. It's real simple. It's taking biological information of any type and directly converting it to electrical information. And that has tremendous power, being able to do that. Now, most of the devices that you would use in this capacity are going to be nanofabricated. So they're going to be based on silicon or some other metal property. So that makes them very small, makes them very cheap to fabricate. Because of their small size, they have a lot of advantages. But by doing this directly, you get rid of labels. And a common label is a fluorescence label. Well, the problem with fluorescent labels is the one you have to make the fluorescent label. You have to incorporate the fluorescent label in order to measure it, and then it involves some kind of excitation like lasers and so forth. So you make something much more complicated, and sometimes it's actually very difficult to get the label into the right place. And, but that's what's used today. So fluorescence is highly used in biology, kind of replacing radioactivity of the past. But I think the next phase is to get rid of that altogether and be label-free. We're just in that mode of being able to do that. This is mostly involving electrical engineers. But with all this activity, you can get very rapid results, and that's sometimes quite important. Part of that rapid results is because of very high sensitivity. And so, for example, in some types we've looked at some kind of assays that have to run overnight with standard assays, where we can get it done in 5 seconds, if it's direct. There's also, because it's miniaturized, allows you to do many, many assays simultaneously. And these also, because of the nanofabrication, there's low power. And that often allows a number of unique things to be done. And because of the high sensitivity, some of them will even work at the single molecule level. My guess is that's going to greatly increase in the future. Now, I'm not showing you exactly what these are, but this whole concept, I think, is going to be happening a great deal. They're small enough and low-powered enough that I suspect that we're going to see these little devices being made and, for example, being put into the bloodstream or being embedded and collecting information about people and relaying that information, say, to your cell phone or something. So this is a future, a direction I think a lot of activity will happen in. This is our first attempt to do this at Stanford. And it's where we're trying to get a direct electrical detection of a reaction DNA synthesis. And the reason to do that is so that you could actually sequence DNA. So this is a CMOS plate here. We surface coat it so we can put molecules on it. We put a DNA molecule on that. And then we carry out a reaction with the DNA polymerase. And the polymerase will then put a base in extending this reaction. When it puts a base in, the DNA has negative charges. So it adds another negative charge. And it also adds, there's a hydrogen ion being generated in that chemical reaction. That hydrogen ion can diffuse away rapidly. The negative charge cannot diffuse away. So you get a transient negative signal induced, and that gets induced and then it's amplified in the circuitry below. And so you see a little pulse because you added the base. The fact you added that base and you get the pulse, you know the identity of that particular base because you put them in one at a time. Now, this was discovered and developed in the lab in the year 2000. However, it didn't get used. And it didn't get used until it was licensed by Ion Torrent in 2010, 10 years. And it was sitting there for 10 years. And this was a conceptualization problem for industry, not the lab. We understood that it would work, but it would require substantial investment and effort to make it work at a commercial level. And it really took, that 10 years was because at this stage, people said, why do you need DNA sequencing? And we have capillary electrophoresis by ABI. And why would you want to displace that? In fact, we tried to sell this to ABI and said, we don't need it. We have capillary electrophoresis. And so there's a paradigm shift here to happen, but that paradigm shift was in fact resisted by industry. So I think the same kind of thing is happening, and will happen over and over again, is the feeling, oh, it's of no value, until you realize that it is. So we tried to license this to ABI for a few $1,000s. They did not want it, so we let it sit there. Ion Torrent took it. In fact, some of my students went to Ion Torrent and helped develop the technology. It then sold for over $700 million. So there was a lot of value here, but it was hidden and not recognized. And I think that's often the case of some of these technologies.
Good morning. My name is Gale Norton, and I'm here to talk a little bit about regenerative medicine. Stem cells, the power to cure diseases. About 15 years ago, if you asked the average population what's a stem cell, probably very few would be able to tell you. That's all changed. A lot of that has been changed in California by the proposition a few years ago that approved a multi-billion-dollar grant to establish the California Institute for Regenerative Medicine. So stem cells really do have tremendous power. Probably what's been the most successful are bone marrow transplants. We've all heard of them. Yet since the mid-'80s, even with so much university work and so many companies focused on cell-based therapies, worldwide there have been less than 12 products approved and available now on the market. Why? Well, a number of reasons. Cells have a logistic problem. They have a set shelf life outside of the body. In addition, there's no format for being able to upscale, make lots and lots of cells to be able to go and treat millions of patients. But we've been addressing that. But one of the things that really has affected the field is that there was poor reimbursement for many of these cell-based therapies. And a number of companies in the early 2000s actually went bankrupt. My former company, Advanced Tissue Sciences, had 3 FDA cell-based products, yet we declared bankruptcy in 2002. Finally, the company was reorganized and bought by Shire for $750 million a few years ago. So learning from this, we've established Histogen, which is a regenerative medicine company that is not based on cells in the product, but the power of what cells can produce, delivering those chemicals to be able to stimulate your own stem cells in your body. We also are focusing our first products on aesthetics, skincare, and hair growth to address the reimbursement issue so that we can build a powerful platform to be able to develop a series of therapeutic products.
So I'm Harry Staley. I'm with Progenity. What I'm going to do is take you through a story, if you like, that some of you may be familiar with, but in the area of genetic testing and how sequencing and these various technologies have evolved. So prior to 1997, there was a tremendous effort looking at fetal cells to see if we could develop non-invasive technologies for the determination of Down syndrome and various other genetic disorders. That was decades and billions of dollars, and it basically proved to be a forced start. In 1990, and we're continuing at it, so I'll come back to fetal cells in a second. In 97, Dennis Lowe discovered circulating cell-free fetal DNA. And this is bits of DNA that come from the fetus and the placenta into the maternal circulation. And that proved very intriguing, because here was another non-invasive source. The alternatives were things like amniocentesis or chorionic villi sampling, or probabilistic methods involving various serum proteins and nuchal translucency, which is a form of ultrasound. Not very satisfying, but sort of effective. Now, the challenge was actually quite formidable. So from 97 In 2005, I licensed the technology for cell-free from Oxford University, 2005. And then we set about looking at ways of being able to detect it. Now, the challenge is quite formidable. There's a 10% cell-free fetal DNA floating around in a maternal background. Now, remember, 50% of the fetal DNA actually comes from the mother, okay? So you've got a very complex situation. So if you were trying to determine chromosome 21, which is the trisomy that's responsible for Down syndrome, you need to quantify it. You're trying to measure a 1.05 greater amount of fetal DNA over a baby, over a euploid pregnancy, a normal pregnancy. So an extraordinary challenge. Now, of course, there's another dimension to this. 10% cell-free is not truly representative because we're all different, and you could have a range from 2% to 20% cell-free. And obviously, the problem becomes even greater the lower you go. So those were the challenges that we faced. Now, in 2008, we had a breakthrough with the advent of next-gen sequencing. We now had hope that we would be able to detect these disorders from the mother's blood. And that was realized, and in 2011, we actually launched, the very first test was launched onto the marketplace. It's the most rapid adoption of a genomic test that's commercial, that has a real clinical benefit in the history of genetic tests. Now, going on, okay, we've since added microdeletions. These are tiny bits of chromosomes that switch location from one place to another and cause hundreds of potential diseases. And that was only recently done by a number of players. Now, moving further, what is the big challenge? We still need probably 2 to 5 years before we're able to actually look at single mutations, polygenetic diseases, which will really, really be the revolution in this field. And again, that's an extraordinary challenge, but with the advent of next-gen sequencing, we're quickly making progress towards achieving that goal. Now, the elusive holy grail remains fetal cells. And the challenge with fetal cells, the advantages of fetal cells are obviously, for me, very, very powerful because you can get large segments of DNA as opposed to these tiny bits that you have to reassemble. The challenges are that there are very, very few fetal cells, and it's been extraordinarily difficult to scale up. But there's still continuing effort in this arena. Now, NIPT, non-invasive prenatal testing, will open a treatment. One of the most exciting things for me is that it's not so much the detection of these disorders, is the fact that if we're able to detect these disorders or these genetic conditions early in the pregnancy, there is a high probability that using stem cells and other approaches, we can go in, or physicians can go in and treat the disease before the baby's born. And that's the exciting, if you like, prospective revolution that we're facing. Good example is congenital adrenal hyperplasia, where if you're able to capture and detect the presence of this disease in girls, disorder in girls, between weeks 7 or 8 of pregnancy, a simple drug can actually cure the child. Okay, that's one of the early well-understood examples. I gave you another example there. Autism appears to develop in the second trimester of pregnancy. Autism is highly treatable the earlier it's caught. Again, this is speculative, right? So I'm not saying we'll be able to do all these things, but once you're able to detect early in the pregnancy, it opens up these opportunities. So here you are. So you now have the technology and the tools that are rapidly emerging that could enable us to intervene very early in pregnancies and potentially rescue the pregnancy and potentially cure diseases in utero. Now, the testing won't stop there, as you're all familiar, and we're reading about it all over the place. I think there'll be genetic testing throughout life because of these epigenomic, the way the environment interacts with our genetic makeup.
Mark, this is Deb Lecom. I'm one of the co-founders and the CTO of Proteus Digital Health. Proteus was founded on the idea of adding electronics to existing therapies to make them more effective. And what we focused on most recently is adding electronics to medicines to make them digital medicines. And the way the system works is that there's a silicon chip literally inside the pill. And when it's swallowed, that turns into a very small communication device that's picked up by a patch that's worn on the torso. And that patch is also a computing sensing platform that measures heart rate, breathing rate, angle of repose when you're sleeping, measures temperature and acceleration, so you know how much walking and activity one is doing. And that communicates with the cloud, ultimately through cell phones, and we have displays that allow people to monitor their performance, and it's shared with their friends and their family, so we can close the loop on taking medicines and also measure the effect of those medicines. So you can think of a very large, big data space in 3 dimensions, one axis being what drugs are actually being taken, not just which ones are prescribed. Another axis being what's the physiologic response to those medicines. And the third axis, since I'm accompanied by all these geneticists, is what's the genome of the various patients. And over time, gathering that very large database may allow us to predict which medicines will be more effective for a given patient population.